Hi everyone. For learning outcome number 4 of module 6, we're going to be talking specifically about the different types of lymphocytes and how they help our immune system combat the pathogens that are able to enter our body. Lymphocytes are going to be the primary cells of the lymphatic system, and they are responsible for specific immunity, which can also be called adaptive immunity. They will respond to the presence of 3 things: invading organisms, such as bacteria and viruses; abnormal body cells, such as if you have a cell that has been infected by a virus or a cancerous cell; and also foreign proteins, such as, for example, toxins that might be released from some bacteria. Now, lymphocytes, they will attempt to eliminate these threats, or actually decide if they're harmless or not, by a combination of either physical or chemical attacks. They will travel throughout the body, circulating in the bloodstream, therefore the blood vessels. Then they will move through the peripheral tissues, and eventually they will return to the bloodstream through the lymphatic system. Now, the time spent within the lymphatic system will vary. It all depends if they encounter a pathogen or not. And if they don't encounter any pathogen, they might stay for a few hours. But if they do, they might stay a few days or even years, depending on the type of disease. Now, regardless of what type of invading pathogen or foreign proteins they encounter, the lymphocytes will respond by initiating an immune response. There are 3 different classes of lymphocytes that circulate in the blood. We have what we call the T cells, and these are thymus dependent. And as we noticed, thymus starts with a T, and that's why they're called T cells. The next type of cells are called B cells, and they are bone marrow derived, so they originate in the bone marrow. And bone marrow starts with a B, therefore these are called B cells. And the NK cells are natural killer cells, so that's where the NK comes from. Now, each time we'll have a distinctive biochemical and functional characteristic, so let's look at them separately. As I said earlier, the lymphocytes, they're going to be the primary cells of the adaptive immune response. Now, there are 2 basic types of lymphocytes. We have the B cells and the T cells, and if we notice from this image, they're actually identical morphologically. They will have this large central nucleus surrounded by a very thin layer of cytoplasm, and they are going to be distinguished from each other by their surface protein markers, which will be the ones that will recognize the pathogens, as well as by the molecules that they secrete. Now, while B cells mature in the red bone marrow, that's why they're called B cells, the T cells, they're going to mature in the thymus, and that's why they're called T cells. But both of them initially developed from the bone marrow. So again, the B cells mature in the red bone marrow, the T cells mature in the thymus, but both will develop from the bone marrow. Now, both B cells and T cells, they're found in many parts of the body, circulating in the bloodstream and in the lymph as well. And they will reside in those secondary lymphoid organs, which we have talked already about, which are the spleen, the tonsils, the lymph nodes. And we will discuss these structures or organs later in this module. B cells are immune cells that will function primarily by differentiating into plasma cells, which are also known as plasmocytes, which are the cells that will produce antibodies, as we can see from this image. An antibody is any of the group of proteins that will bind specifically to pathogen-associated molecules known as antigens. Therefore, an antigen is a chemical structure on the surface of a pathogen that binds to either T or B lymphocyte antigen receptors. Therefore, what needs to happen first is that an antigen from a pathogen will bind to the B cell receptors, which are located on the surface of these cells, and this will activate the B cells to differentiate into plasma cells, which will then secrete a specific soluble form of their surface antibodies that will target the antigens. With regards to the T cells, they do not secrete antibody, but they will perform a variety of functions in the adaptive immune response because there are several different types of T cells, and they will have the ability to either secrete those soluble factors that will communicate with other cells of the adaptive immune response, or they will go and destroy the cells infected with intracellular pathogens. And as we can see from this image, there are two main types of T cells, which are the cytotoxic T cells, which are also known as CD8 positive T cells, and the helper T cells, which can also be called CD4 positive T cells. And the last type of lymphocyte, which are the NK cells, or natural killer cells, they're going to be very important because they will not only participate of the innate immune response like we saw, but they will be circulating in the blood, and they will also contain certain cytotoxic granules, where basically means that they're going to be killing cells, right? Because cyto is cell and toxic means that it's killing. So they're going to function to kill and destroy cells that have been infected. So it does share this same mechanism with the cytotoxic T cells of the adaptive immune response. Now, NK cells, they're going to be among the body's first lines of defense against viruses and certain types of cancer. And this is just a little review of the main types of lymphocytes in the adaptive immune response. So let's look at them in a little bit more detail on the next few slides. We have approximately 80% of the circulating lymphocytes being classified as T cells. As we talked already about the T cells, they will originate within the bone marrow, but then they will migrate to the thymus to become activated. Another word for this is immunocompetent. So once they get to the thymus and they mature, they become immunocompetent. And there are several different types of T cells. I have some listed here, which are the main ones. We have the cytotoxic T cells, which we have talked already about. These are going to be also called T killer cells or CD8 positive T cells. Next, we have the helper T cells, which can also be called the CD4 positive T cells. The regulatory T cells, which can also be called suppressor T cells, memory T cells, and naive T cells. So let's look at their function separately on the next few slides. Let's start with the cytotoxic T cells. These lymphocytes, they're going to be responsible for providing what we call a cell-mediated immunity, which basically is telling us exactly this, that these cells, they will attack and destroy foreign cells or body cells infected by viruses by a direct contact of the cytotoxic T cells with the infected cells. And this is why they also call these cells T killer cells, right? And they will kill target cells by inducing apoptosis, which as we know is programmed cell death, using the same mechanisms as the natural killer cells. So they will express the transmembrane Fas ligand, which will bind to the Fas molecule on the target cell. And once the two cells are bound, the cytotoxic T cell will release perforins and granzymes that are contained in their cytoplasmic granules. Now, perforin is a glycoprotein that's responsible for pore formation in the cell membrane of the target cells, and this will lead to the formation of little channels in the target cell membrane, and this then causes the cell to die. And granzymes are serine proteases Therefore, enzymes, like the name says, granzymes, that will also induce apoptosis of the target cells. The best thing that could happen to our body is to have a cell that is really good at killing a virally infected cell before the virus can complete its replication cycle, as this will result in the production of no infectious particles. And as more cytotoxic T cells are developed during an immune response, they will actually overwhelm the ability of the virus to cause a disease. And in addition, each of these cytotoxic T cells, they can kill more than one target cell, making them especially effective. Now, the cytotoxic T cells are so important in the antiviral immune response that some actually speculate that this was the main reason the adaptive immune response evolved in the first place, to be able to protect us from the viral infections. Therefore, as we take a look at this image, we can see that a virus will target a healthy cell, which then becomes infected with this virus. The T cells, or the cytotoxic T cells that are circulating in the bloodstream, they will recognize this infected T cell. They will then secrete what we call cytokines, which are any of a number of substances like interferon and interleukin, or even growth factors, which are going to be secreted by these cells. This will in turn activate the replication of more of these cytotoxic T cells. And these new T cells will, like I said earlier, bind to the target cells and release certain chemicals like perforin and granzymes that will lead to the apoptosis of the target cell, and therefore elimination of the virus inside of our body. And the debris of this cell that just suffered apoptosis will then be cleaned up by phagocytic cells of the innate immune response, such as macrophages and neutrophils. Next, we have the helper T cells, which are the ones that will bear the CD4 molecule, so that's why they're known as CD4 positive T cells as well. And they will function by secreting certain cytokines that will act to enhance other immune responses. And this is why they're called helper cells, because they will help with the immune response. And there are two classes of helper T cells. These are going to act on different components of the immune response. These cells are not going to be distinguished by their surface molecules because, like I said, both will express the CD4 molecule, but they will be distinguished by the characteristic set of cytokines that they secrete, and these cytokines are the ones that will determine which cells they will help. The first type of cell are what we call the Th1 cells, and these are a type of helper T cells that will secrete cytokines that will regulate the immunological activity and the development of a variety of cells that will include macrophages and even other types of T cells. And these cytokines that are present in the Th1 cells are what we call interferon gamma, IL-2, and lymphotoxin alpha. So let's see how this process occurs using this image over here. So as we can see, there's going to be a virus that's going to be circulating in the bloodstream, which will be detected by a macrophage, which is a cell of the innate immune response. Remember that the innate immune response is the first one to act, as it gives time for the adaptive immune response to create all these steps that we will see now to destroy the virus in this example. Next, this macrophage will present the virus antigen to the Th1 cell, and this will make the Th1 cell release certain cytokines. In this case, they are showing an interleukin, which more specifically is probably interleukin 2. In addition, these cells can also secrete, like I said, interferon gamma and lymphotoxin alpha that will then recruit your cytotoxic T cells to destroy the infected cells and also to replicate and give rise to more cytotoxic T cells to destroy the same virus that might be circulating in our body. The other type of helper T cells are what we call the Th2 cells, and on the other hand, these are going to be cytokine secreting cells that will act on B cells to drive their differentiation into plasma cells to make more antibody. In fact, the T cell help is going to be required for antibody response to most protein antigens. So let's use this image to see how this will occur. So you're going to have a pathogen, in this case can be a virus, that will be grabbed by the macrophage, which will then release interleukin 1, which will recruit the Th2 helper T cells to secrete interleukin 4 and interleukin 5, which will then activate the B cells to proliferate. And when you have more B cells, you're going to have more plasma cells, which are the ones that produce the antibodies. Therefore, you're going to have more antibodies circulating to be able to destroy these invading pathogens. The regulatory T cells or suppressor T cells are the most recently discovered of the types listed here. So, of course, there's less that we know about them. In addition to CD4 on their surface, they will bear the molecules CD25 and FOXP3. Now, exactly how they function is still under investigation, but it is known that they suppress other T cell immune responses. So basically, they work as a control of the immune response. In other words, they're creating a maintenance of homeostasis because they're trying to make sure that there's not too many of these T cells circulating, so they will suppress the immune response of some of the T cells. And in this way, they also maintain what we call the self-tolerance by either inhibiting T cell proliferation or inhibiting cytokine production. And by doing this, they will prevent certain types of autoimmune diseases that might happen if you have too many T cells circulating. And this will also limit chronic inflammatory diseases due to an excess presence of T cells within the circulatory system. A memory cell is a variety of both B and T cells that forms after you have an exposure to a pathogen. Therefore, when you're re-exposed to the same pathogen, your body will be able to mount a rapid response because you already have those memory cells circulating throughout your body. And the most important fact is that unlike other leukocytes that have a short lifespan, the memory cells, they live for many years. The other interesting fact about the memory T cells is that they do express certain chemokines and adhesion receptors that will allow them to extravasate into non-lymphoid tissues, and this is how they're able to live for a longer period of time. Memory T cells are necessary for protective immunity against invading pathogens, especially under conditions of immune suppression, so individuals that lack a lot of the immune response. However, their presence also threatens the transplant survival, making transplantation a great challenge. Because let's say a patient is receiving an organ donation and their memory T cells might recognize an antigen from the organ that's being donated and want to destroy the cells of this donated organ. So this is why it can be challenging for transplantation. As we know, the lymphocytes, they will develop and mature in what we call the primary lymphoid organs, which can be the bone marrow and the thymus, but some they will mount their immune responses from secondary lymphoid organs. Now, a naive lymphocyte is one that has left the primary organ and entered a secondary lymphoid organ. However, the naive lymphocytes, even though they're fully functional immunologically, they have not yet encountered an antigen to respond to. Therefore, we can actually say that they are opposite of the memory cells, which are the ones that have encountered already, and they stay in alert for any pathogen that wants to reinfect the body. So like I said, these naive T cells, they will concentrate in the secondary lymphoid organs, which includes the lymph nodes, the spleen, and the lymphoid nodules. And all of these tissues, they have many features in common, including the following: the presence of what we call a lymphoid follicle, which is the site of formation of lymphocytes, with specific B cell rich and T cell rich areas, which we will cover more when we talk about the lymph nodes. They will have an internal structure of reticular fibers that are going to be associated with fixed macrophages. They will have germinal centers, which are the sites of rapidly dividing and differentiating B lymphocytes. And they will also have what we call a specialized postcapillary vessel known as high endothelial venules. And the cells that are lining these venules, they're going to be thicker and more columnar than normal endothelial cells, which will then allow cells from the blood to directly enter these secondary lymphoid organs or tissues. Now, because the naive T cells, they lack these homing receptors for peripheral tissues and also the chemokine receptors for this inflammatory cytokine, they are unable to enter the non-lymphoid tissues. Therefore, they can only be present in the secondary lymphoid organs and tissues. Now that we're done with the T cells, we're going to focus on the B cells, which originate and become immunocompetent within the bone marrow. And they will account for 1 to 15% of the circulating lymphocytes. When stimulated by the exposure of an antigen, the B cell, they can differentiate into plasmocytes or plasma cells, and these cells are going to be responsible for the production and the secretion of antibodies. These soluble proteins, which are the antibodies, they will react with specific chemical targets that are called antigens, just like the T cells. And antigens are usually associated with pathogens, parts or products of pathogens, or other foreign compounds. And when an antibody binds to its corresponding antigen, then this starts a chain of events leading to the destruction, the neutralization, or even the elimination of the antigen. Antibodies are also known as immunoglobulins, which we have covered already. And because the blood is the primary distribution route for immunoglobulins, B cells are said to be responsible for antibody-mediated immunity, or also called humoral immunity, where humoral means liquid immunity because it's present in the blood. Now, memory B cells, they're going to be produced by the division of these activated B cells, and activation occurs following the exposure to a particular antigen. We do have memory B cells that will become activated only if the antigen appears in the body at a later date. And this mechanism is similar to the mechanism for the memory T cells that we have already talked about. So let's see how this mechanism occurs. Basically, you're going to have a virus circulating in the blood, and it will infect a healthy cell. Then it becomes an infected cell, which will bind and find a B cell. So the B cell will bind to the antigen present on the cell-infected cell, and it will release certain cytokines, mainly interleukin, and this will activate B cells to divide several times, and this will create a memory B cell as well. And the B cells will then give rise to the plasma cells or the plasmocytes, which are the ones that produce antibodies. And then the antibodies will send a signal to macrophages, for example, for them to go and destroy the virus. And lastly, we have what we call the NK cells or natural killer cells, and they will represent the remaining 5 to 10% of the circulating lymphocytes. These lymphocytes, they will attack foreign cells, normal cells that are infected with viruses, and cancer cells that appear in normal tissue. And on the image, we can see a cell that is infected by a virus. The NK cell will recognize the antigen of the virus by activating the receptor that will send a signal to the infected cell for it to die by apoptosis, basically for the cell to kill itself, right? And this process is similar to what occurs in the cytotoxic T cell mechanism, which we have already discussed. Now, this continual policing of the peripheral tissues by the NK cells and the activated macrophages, it has been called the immunological surveillance. So these cells are basically the detectives of our immune system. Now, these cells, they originate in the bone marrow, but they're also found in lymph nodes, in the thymus, liver, and in the uterus. In addition, some studies have shown the presence of NK cells in the placenta, and this leads them to believe that NK cells have an important role during pregnancy.